Let's begin with something that seems simple, but is actually surprisingly deep. Hair is not a living thing in the way your heart or liver is living. That might sound strange at first, because hair clearly grows, and anything that grows feels alive. But what is really happening is more like a production process happening under the skin. Each strand of hair comes from a tiny structure in your scalp called a follicle. You can think of a follicle as a microscopic factory. It sits in the skin, reaches down into tiny blood vessels, and takes in raw materials from your body, like proteins, fats, oxygen, and energy. Then it uses those materials to build a long, thin fiber that pushes outward through your skin. That fiber is what you see as hair. Now here is a key idea that changes how you think about it. The visible hair strand itself is not alive. Once the hair leaves the follicle, it is basically a hardened biological fiber. It does not repair itself, it does not heal like skin, and it does not respond to signals from your body anymore. It is already finished material, like something that came out of a 3D printer. So when people talk about hair loss, they are not talking about damaged hair that needs healing. They are talking about a change in the behavior of the follicle, the factory, not the product. If the factory slows down, shrinks, or stops working, the hair becomes thinner, shorter, or disappears entirely. The problem is not the hair strand you can see. The problem is the invisible machinery underneath it. This leads to an important shift in thinking. If you want to understand hair loss, you should not stare at the hair itself. You should ask, what is happening to the follicle that produces it? Now imagine each follicle as a tiny machine that runs in cycles. It does not produce hair continuously forever. Instead, it goes through phases. First, a growth phase where it actively builds hair. Then a rest phase where activity slows down. Then, in some cases, a shedding phase where the old hair falls out and a new cycle begins. At any given moment, different follicles on your head are in different phases of this cycle. That is why your hair does not all fall out at once. It is a staggered system, like thousands of small clocks all ticking at different speeds. But here is where things become more interesting and more complicated. These follicles are not operating independently in a vacuum. They are constantly receiving signals from the rest of your body. Hormones carried in your blood, local chemical signals in the skin, genetic programming inside the cells, all of these influence whether a follicle stays active, slows down, or shrinks over time. So when we say hair grows, what we really mean is that a follicle is successfully continuing its production cycle under the influence of a very complex set of internal instructions. And when we say hair is thinning, we often mean that the follicle is gradually producing thinner and shorter strands until, in some cases, it produces almost nothing visible at all. This is why hair loss is not a simple on-off switch. It is more like a gradual reduction in output from a factory that is still technically present, but becoming less and less efficient over time. The machinery is still there, but the performance is changing. And once you see it this way, you start to realize something important. Hair is not just a cosmetic feature. It is a visible output of invisible biological decisions happening constantly under your skin. Let's continue by looking at the chemical signal that is most often discussed in relation to male pattern hair loss. Inside the human body, hormones act like messengers. They travel through the bloodstream and carry instructions to different tissues. One of the most well-known hormones involved in hair biology is testosterone. But testosterone is not the end of the story. In certain tissues, an enzyme can convert testosterone into a slightly different molecule called dihydrotestosterone, often shortened to DHT. Now, instead of thinking of DHT as a villain, it helps to think of it as a modified version of a familiar signal. The body is not producing good and bad hormones in a moral sense. It is producing chemical variations that have different effects depending on where they land and what cells receive them. Here is a useful analogy. Imagine testosterone as a general radio broadcast sent across the entire country. Most places receive it in a standard, stable way, but in some regions, there is a machine that slightly alters the signal before it reaches local receivers. That altered version is DHT. It is not completely different, but it has a different intensity and different impact depending on the receiver. Now, each hair follicle can be thought of as a receiver tuned to these hormonal signals, but not all receivers are identical. Some follicles are highly sensitive to DHT. Others are relatively resistant. This difference is not random in the moment. It is strongly influenced by genetics, meaning it is largely determined long before hair loss begins. When a follicle is exposed to DHT and is sensitive to it, something interesting happens over time. The growth phase of the hair cycle becomes shorter. Instead of producing thick, strong hair for a long period, the follicle starts producing thinner, weaker hair for shorter periods. This process is gradual. It does not happen overnight. It can take years. Think of it like a factory slowly receiving less efficient instructions. The machines still work, but each production cycle becomes slightly less effective. The output does not stop immediately. It degrades step by step. First, the product becomes thinner, then shorter, then less visible. Eventually, in some follicles, production becomes so minimal that it appears as if the factory has shut down completely, even though it still exists under the skin. This is why hair loss patterns often follow recognizable shapes, especially in men. It is not because hair follicles in those areas are fundamentally different in structure. It is because those follicles tend to be more sensitive to the same hormonal signal. The signal is distributed throughout the scalp, but the response is not uniform. However, there is an important scientific caution here. Even though DHT is strongly associated with this type of hair loss, we do not fully understand why follicle sensitivity varies so much between individuals. We can measure correlations, we can observe patterns, we can even modify DHT levels and see effects on hair in some cases. But the full chain of cause and effect, from gene expression to follicle behavior, is still an active area of research. So what we are left with is a partial picture. We know DHT plays a significant role, we know follicle sensitivity matters, we know genetics sets the stage. But we do not have a single, complete explanation that accounts for every variation we see in real life. And that uncertainty is important. It reminds us that biology is not a simple machine with one control knob. It is a layered system where many signals interact and where the same chemical can produce different outcomes depending on context, location, and time. Now we come to the idea of oils and the claim that certain natural substances can block DHT and restore hair growth. This is where science and storytelling often begin to drift apart, so it is useful to slow down and examine what that phrase actually means. When someone says an oil blocks DHT, they are usually implying a very specific biological action, that the oil somehow reduces the amount of DHT in the scalp or prevents DHT from interacting with hair follicles. On the surface, that sounds simple, but in biology, blocking is not a single mechanism. It can refer to many different processes that only partially overlap. For example, a substance might reduce the activity of the enzyme that converts testosterone into DHT. That would be one kind of blocking. Another substance might not change DHT levels at all, but could reduce inflammation in the scalp, which indirectly changes how follicles respond to hormones. Yet, another substance might improve skin hydration or circulation, making hair appear thicker or healthier without changing hormone signaling in any meaningful way. So before accepting the idea of a DHT blocking oil, a scientist has to ask a more precise question. What exactly is being blocked? Where in the body is it being blocked? And how strong is the effect compared to known biological pathways? This is where things become complicated. The scalp is not a simple surface. It is a living tissue with blood vessels, immune cells, nerves, glands, and thousands of hair follicles all interacting at once. Any substance applied to the skin has to pass through multiple biological barriers before it can meaningfully affect hormone activity inside follicles. Many molecules simply do not penetrate deeply enough, or they are broken down before they can have a strong effect. That does not mean topical oils are useless. It means their effects are usually indirect and often modest. For instance, some oils may reduce dryness or scaling of the scalp. A healthier scalp environment can reduce hair breakage or improve the appearance of density. Some oils may have mild anti-inflammatory properties, which could slightly influence follicle behavior in sensitive individuals. But these effects are not the same as directly reprogramming the follicle's response to DHT. Another important point is how easily perception can be mistaken for biological change. Hair is highly sensitive to visual conditions. Lighting, hair length, styling direction, and even moisture can dramatically change how dense hair appears. When someone starts using an oil, they often pay closer attention to their hair, change their grooming habits, or combine multiple changes at once. Any of these can create the impression of improvement, even if the underlying follicle biology has not significantly changed. This is where controlled scientific studies become important. In proper clinical research, variables are isolated. One group receives the treatment, another does not, and neither group knows which they are in. When oils are tested under such conditions for significant hair regrowth or strong DHT suppression, the results are often inconsistent or modest compared to established medical treatments that directly target hormonal pathways. So the careful scientific position is not that oils have no effect, but that their effects are usually subtle, context-dependent, and not reliably comparable to pharmaceutical interventions designed specifically to alter hormone activity. And this leads to a broader lesson that goes beyond hair. In complex biological systems, it is very easy to describe something in simple language like blocks X. But reality is rarely that clean. Most interventions do not block anything completely. They shift probabilities, adjust local environments, and produce small changes that may or may not add up to visible outcomes, depending on the individual system they act on. One of the most interesting parts of this topic is not hair itself, but how our minds try to understand it. Human beings are extremely good at finding simple cause and effect relationships. In everyday life, that skill is powerful. If you touch a hot surface and get burned, you quickly learn to avoid it. If you eat something and feel sick, you connect the two events. This kind of thinking is efficient and often correct in simple systems. But biology is not a simple system. A hair follicle is not like a light switch where one input produces one predictable output. It is more like a crowded control room with many knobs being adjusted at the same time. Hormones are changing. Local inflammation is rising or falling. Blood flow varies slightly from moment to moment. Genetic programming defines baseline sensitivity. Even mechanical factors like brushing or tension can influence behavior over time. When many variables interact like this, the brain still tries to simplify the story. It prefers explanations like, this oil blocks that hormone, so hair grows back. That kind of explanation feels satisfying because it compresses complexity into a single mechanism. But the danger is that compression can remove important information about how the system actually behaves. To understand why this is difficult, imagine trying to predict the weather by looking at only one variable, like temperature. Temperature matters, but it is not enough. Wind, humidity, pressure systems, ocean currents, and geography all interact. If you only focus on one factor, your predictions will sometimes be right and sometimes completely wrong, without a clear pattern. Hair biology is similar. DHT is important, but it is only one part of a larger system. Focusing only on DHT is like trying to understand a storm by measuring only the temperature of one city. Another reason this topic becomes confusing is timescale. Many biological changes happen slowly. Hair follicles operate in cycles that can last weeks or months. If a change slightly improves follicle activity, the effect might not be visible immediately. It may take months before differences become noticeable, and by that time many other factors may have also changed. This makes it very difficult to isolate cause and effect in everyday observation. There is also the issue of confirmation bias, which is a natural feature of human cognition. If someone expects an oil to work, they will tend to notice improvements and overlook non-improvements. Hair is especially vulnerable to this because small visual changes can look significant depending on lighting, angle, or styling. A slight reduction in frizz or dryness can be interpreted as new growth, even if the number of active follicles has not changed. This does not mean people are irrational. It means the system we are trying to interpret is subtle, and our perception tools were not designed for microscopic biological measurement. Scientists deal with this by using controls, measurements, and statistical comparisons. Instead of asking, does it look better, they ask, does it consistently produce measurable change across many subjects under controlled conditions? This approach often removes illusions created by short-term observation. So the difficulty in explaining hair loss treatments is not just a lack of knowledge. It is a combination of system complexity, slow timescales, overlapping variables, and the natural tendency of the human mind to prefer simple stories over layered explanations. And when you put all of that together, you begin to see why even well-intentioned discussions about hair biology can easily drift away from what the underlying science actually supports. Now we reach a deeper question that sits underneath the science. Why is hair loss so difficult to fully understand and explain, even with modern biology? At first glance, it might seem surprising. We know what hair is made of. We can identify hormones involved. We can even influence hair growth with medications in some cases. So why isn't the picture complete? The answer is that hair growth is not controlled by a single system. It is the result of multiple biological layers interacting at different scales of time and organization. At the smallest scale, you have genes inside cells. These genes determine how sensitive a follicle is to hormones like DHT, how long its growth phase lasts, and how it responds to stress signals. But genes are not fixed instructions that always behave the same way. They are more like conditional programs that turn on or off depending on the environment inside the body. Above that level, you have chemical signaling systems, hormones, inflammatory molecules, and growth factors circulating through blood and tissue. These signals are constantly changing based on sleep, nutrition, stress, age, and overall health. A follicle is not reading one signal. It is integrating many signals at once, like a constantly updating calculation. Then there is the tissue environment itself. The scalp is not just passive skin. It contains immune cells that can trigger inflammation, blood vessels that regulate nutrient delivery, and structural changes that occur gradually over time. Even small changes in blood flow or local inflammation can shift how a follicle behaves over long periods. And finally, there is time. Hair follicles operate in cycles that are slow compared to many other biological processes. A change that begins today might not fully show its effect for months. That delay makes cause and effect extremely hard to trace in real life. By the time you notice a difference, many other variables may have also changed. When you combine all of these layers, you get a system that behaves more like a living network than a simple machine. And networks like this are notoriously difficult to predict. Small differences in starting conditions can lead to very different outcomes over time. This is one reason why two people with similar hormone levels can experience very different patterns of hair loss. Another difficulty is that we are still missing parts of the full picture. We know many important mechanisms, but not all of them. For example, we do not fully understand why certain follicles in specific regions of the scalp are more genetically sensitive to DHT than others. We also do not fully understand why some follicles remain stable for decades despite similar hormonal exposure. Science progresses by building models that explain as much as possible with available evidence. But in complex biological systems, these models are always incomplete. They are refined over time, not finalized in a single step. This is why different treatments for hair loss can appear inconsistent. Some people respond well, others less so, and sometimes results change over time. It is not always a matter of something working or not working, but of partial effects interacting with individual biology in unpredictable ways. So the difficulty is not just in finding a single missing answer. The difficulty is that the system itself is layered, adaptive, and sensitive to small variations that are hard to measure in everyday life. And that brings us to an important realization. In biology, understanding often means accepting complexity rather than eliminating it. Now let's step back and ask a more uncomfortable but important question. What do we actually mean when we say something restores hair? In everyday language, restoration sounds simple. It suggests that something is returned to a previous state, like repairing a broken object or refilling a container that was emptied. But biological systems do not always work in reversible ways. A hair follicle is not a mechanical part that can be cleaned and reset like a clogged pipe. It is a living structure that changes over time. As it ages or becomes less active, it can shrink, produce thinner fibers, and eventually enter a state where its output is minimal. This process is gradual, and importantly, not always fully reversible. That leads to a key scientific distinction. Improving appearance is not the same as restoring original biological function. A treatment might make hair look thicker by increasing strand diameter, reducing breakage, or improving moisture content. It might even slightly extend the growth phase of existing follicles. But that does not necessarily mean dormant or miniaturized follicles are being fully revived. This is where interpretation becomes tricky. Human perception is very sensitive to visual density. A small change in light reflection, hair texture, or styling direction can create a strong impression of improvement. Our visual system is not measuring follicle activity. It is interpreting patterns on a surface. That gap between biological change and visual perception is where many misunderstandings begin. There is also the issue of baseline fluctuation. Hair density is not perfectly constant from day to day. It naturally varies with washing frequency, humidity, hair length, and even how hair is combed. When someone starts using a new oil or treatment, they are often paying much closer attention than before. This alone can make small normal variations feel like meaningful progress. From a scientific perspective, the only reliable way to determine true restoration would be to measure follicle activity directly over time under controlled conditions. That means tracking hair counts, growth rates, and follicle size across many subjects, while controlling for external variables. In such studies, many interventions that seem impressive in casual observation show much smaller or inconsistent effects when measured rigorously. This does not mean that change is impossible. It means that biological change exists on a spectrum. Some interventions have strong, measurable effects, especially those that directly target hormonal pathways or cellular signaling mechanisms. Others produce weaker, indirect, or highly individual responses that are harder to generalize. So when we hear claims about fast restoration, it is important to translate that language into biological terms. Fast compared to what baseline? Restoration of what exactly? Strand thickness, follicle count, growth rate, or cosmetic appearance? The more precisely we define the claim, the narrower the set of mechanisms that could realistically support it. And this is where science becomes less about belief and more about translation. We take a vague, emotionally appealing statement and convert it into measurable biological questions. Often, that translation reveals that the original idea was mixing several different processes together into one simple phrase. So the challenge is not only understanding hair biology, it is also learning how to separate what we see, what we assume, and what is actually being measured inside a living system that is constantly changing. Now let's bring everything together and look at a final, deeper question. What does hair loss teach us about how reality itself behaves when we study complex living systems? At first, hair loss seems like a narrow biological problem. A few hormones, a few follicles, a visible change on the surface of the body. But when you look more carefully, it becomes a small window into something much larger, the limits of simple explanation. In physics, we are used to systems that can be described with clean equations. If you know the forces, you can often predict the motion. That works beautifully for planets, projectiles, and many engineered systems. But biology is different. It is not just a collection of parts. It is a collection of parts that adapt to each other, respond to history, and change their behavior based on context. A hair follicle today is not exactly the same as it was last year, even if it looks similar. It has been influenced by thousands of small chemical events, cycles of growth and rest, variations in blood supply, immune activity, and genetic expression that turns up or down depending on conditions. It carries a kind of biological memory, not in a conscious sense, but in the way its structure and behavior are shaped over time. This is why reduction to a single cause often fails. When people say it is just DHT, or it is just aging, or it is just inflammation, they are trying to compress a multidimensional system into a one-dimensional story. That compression is useful for communication, but it is not always faithful to the underlying reality. The deeper truth is that hair growth is an emergent behavior. That means it arises from many interacting parts, rather than from a single controlling switch. Emergent systems are difficult to predict because small changes in one part can ripple through the network in nonlinear ways. A slight change in hormone sensitivity might have no visible effect in one person, but a large effect in another, due to differences elsewhere in the system. This is also why interventions can appear inconsistent. A treatment might help one person noticeably, do very little for another, and produce only subtle changes in a third. From a purely superficial perspective, that can look like randomness. But from a systems perspective, it reflects underlying differences in how the network responds to the same input. One of the most important lessons here is epistemological, meaning it is about the limits of knowledge itself. We often assume that if we understand the parts, we automatically understand the whole. But in complex biological systems, understanding the parts is necessary but not sufficient. You also need to understand how the parts interact over time, and even then, there are limits. Measurement tools have resolution limits. Experiments have constraints. Human observation is biased toward visible outcomes. So our models of reality are always approximations, not final descriptions. Hair loss, in that sense, is not just a cosmetic issue or a hormonal issue. It is a reminder that nature does not organize itself around human simplicity. It follows its own structure, which is layered, adaptive, and sometimes resistant to clean explanation. And perhaps the most honest conclusion is this: science does not give us final stories. It gives us progressively better maps. Some maps are detailed enough to navigate with confidence. Others are rough sketches that still miss important terrain. The map of hair biology is somewhere in between, improving steadily but not complete. So, when we look at something as ordinary as thinning hair, we are actually looking at a deep principle of nature itself. Reality is not required to be simple. It is only required to be consistent. And our job as observers is not to force it into simplicity, but to learn how to think clearly, even when the system refuses to be simple. Let's end by asking the most important question that quietly sits underneath everything we've discussed. What should we actually conclude when we look at something like hair loss through a scientific lens? It is tempting to want a final answer, a clear yes or no, a definitive statement about oils, hormones, or treatments. But science rarely offers that kind of closure, especially in systems as layered as human biology. Instead, what we get is something more subtle and, in a way, more honest. We learn that hair is not just a cosmetic feature sitting on the surface of the body. It is the visible output of a deeply complex biological system that is constantly negotiating between genetic instructions, hormonal signals, immune activity, and environmental influences. Every strand of hair is the result of countless invisible interactions that began long before it emerged from the scalp. When that system changes, when follicles shrink, slow down, or stop producing visible fibers, it is not usually the result of a single broken part. It is the result of many small shifts accumulating over time. Some of those shifts we understand reasonably well. Others we only partially understand, and some we may not fully understand yet at all. This is where the limits of human intuition become important. Our brains evolved to handle everyday scales of experience. Objects moving, people interacting, simple cause and effect. But biology at the cellular and molecular level does not always behave in ways that align with those instincts. It operates in probabilities, feedback loops, and thresholds that are not directly visible to us. So when we hear explanations like "this blocks that and regrows hair fast," we should translate them into a more careful language. What system is being affected? How strongly, under what conditions, and with what level of evidence? Often, that translation reveals that the claim is not necessarily false, but incomplete in a way that matters. At the same time, science also teaches humility. The fact that we do not have a complete explanation does not mean nothing is known. It means knowledge exists in layers. Some layers are solid and well-tested, like the role of hormones such as DHT and pattern hair loss. Other layers are still being mapped, like individual variation in follicle sensitivity or long-term regenerative limits. This layered understanding is important because it changes how we relate to uncertainty. Instead of forcing a binary judgment, true or false, we learn to think in degrees. Well-supported, partially supported, weakly supported, or not yet understood. That way of thinking is slower, but it is also more aligned with how complex systems actually behave. And finally, there is a broader philosophical point hiding inside this biological problem. When we study something like hair loss carefully, we are not just learning about follicles or hormones. We are learning something about reality itself, that it is structured, interconnected, and often resistant to simple narratives. The world does not always organize itself around the way we prefer to explain it. It does not guarantee simplicity. Instead, it offers patterns that can be discovered, but only if we are willing to look past overly clean stories and accept a more detailed, sometimes messy truth. And in that sense, hair loss becomes more than a biological condition. It becomes a small, visible example of a much larger principle. Understanding is not about finding the easiest explanation, but about steadily refining our picture of something that is always more complex than it first appears. If we extend this discussion one final step, we arrive at something that is no longer just about hair or even biology, but about how knowledge itself behaves when we push it against the edges of complexity. One of the most important lessons from studying systems like hair growth is that understanding is never a finished object. It is not something you have in a permanent way. It is something you continuously update. In physics, we sometimes like to imagine that once a law is discovered, it is fixed forever. But in real scientific practice, what we actually build are models. Structured approximations that work within certain limits. Hair biology is a perfect example of this. We have models that explain hormone effects. We have models that describe follicle cycles. We have models for inflammation and tissue aging. Each model captures part of the truth, but none of them captures the whole system on its own. The real behavior emerges when all of them interact. And this is where something subtle happens in human thinking. The mind prefers closure. It prefers to say this is the cause or this is the solution. But the deeper we go into systems like this, the more we realize that closure is often an illusion created by simplifying assumptions. For example, when someone says a certain oil blocks DHT, the statement feels complete. It feels like the problem has been localized and solved. But what is usually missing is the rest of the system. How much DHT is affected, in which tissue layer, for how long, in what concentration, and how individual genetic variation changes the response. Once all of that is included, the simple sentence expands into something much less tidy. This is not a failure of science. It is actually a reflection of its honesty. Science does not require stories to be simple. It requires them to be testable and consistent with evidence. And in complex systems, those two requirements often produce descriptions that are layered rather than compact. There is also an important psychological dimension here. Humans are not just observers of reality. We are pattern-finding systems ourselves. We tend to notice improvements, assign meaning to them, and connect them to whatever change we recently made. This is not irrational, it is how perception works. But it means that personal experience alone is not always a reliable guide in systems where many variables change at once. That is why scientific methods rely on repetition, control groups, and measurement across many individuals. The goal is not to deny experience, but to separate signal from noise when the system is too complex for intuition to handle reliably. If we step back far enough, hair loss becomes a small demonstration of a larger truth about nature. Most interesting systems are not linear, not fully predictable, and not reducible to single causes. They are networks of interacting parts, evolving over time, with outcomes that depend on both structure and history. And in such systems, the role of science is not to provide final answers, but to improve clarity. Each experiment reduces uncertainty slightly. Each model removes some confusion while revealing new questions. Understanding grows, but it never becomes absolute. So the final insight is not about hair at all. It is about how we relate to complexity. Reality does not owe us simplicity. It offers structure, but not always simplicity. And the more carefully we observe it, the more we realize that simple explanations are often just early drafts of a deeper story that still has many layers left to uncover. If we push this idea one step further, we reach a point where the discussion about hair loss stops being about biology alone and starts becoming a lesson about the limits of certainty itself. In science, there is a constant tension between what we can measure and what we think we understand. Measurement gives us data. Understanding gives us interpretation. But those two things are not always perfectly aligned. You can measure changes in hair density, hormone levels, or follicle size with increasing precision, yet still struggle to build a single explanation that feels complete. This is because complex systems do not always compress neatly into human language. We prefer explanations that are short, linear, and intuitive. But biological systems are often non-linear, meaning small changes can produce unexpectedly large effects, and large changes can sometimes produce almost no visible effect. That alone breaks many of our everyday expectations about cause and effect. Hair follicles are especially interesting in this regard because they sit at the intersection of biology, chemistry, and time. They are influenced by fast-changing signals like hormones, slower processes like tissue remodeling, and very slow processes like aging and genetic expression patterns. When different timescales interact, prediction becomes difficult, not because the system is random, but because it is layered. One of the deeper scientific insights here is that explanation is not the same as control. We can understand parts of a system without being able to fully predict or manipulate it in every individual case. For example, knowing that DHT plays a role in pattern hair loss gives us a partial map of the system, but it does not automatically allow precise control over every outcome because individual biological variation introduces additional structure we are still mapping. This is where humility becomes an important part of scientific thinking. Not emotional humility, but structural humility. The recognition that our models are always smaller than the reality they describe. Every model leaves something out. The question is not whether it is perfect, but whether it is useful within a defined range. When people encounter simplified claims about hair regrowth or hormone blocking, the risk is not just in believing or rejecting them, but in mistaking the simplified model for the full system. A simplified explanation can be useful for communication, but dangerous if it replaces deeper inquiry. In reality, most biological interventions work probabilistically rather than absolutely. They shift likelihoods, not certainties. They change distributions, not guarantees. That is why responses vary between individuals and why long-term outcomes often differ from short-term impressions. If we extend this way of thinking beyond hair, we see a general principle. Nature rarely operates in binary categories that match human language. It operates in gradients, feedback loops, and overlapping systems. Our language tends to simplify those gradients into categories like works or doesn't work, but reality continues to behave in a more continuous way underneath. So the deeper lesson is not about any specific treatment or mechanism. It is about how understanding itself evolves. At first we create simple explanations, then we refine them as contradictions appear. Then we build more complex models that include exceptions. And even those eventually get replaced or expanded as new evidence arrives. Science, in that sense, is not a collection of final answers. It is a structured way of correcting oversimplified ones. And if there is a final takeaway, it is this. The goal is not to force the world into simple explanations, but to train ourselves to remain comfortable in the space between clarity and complexity, where understanding is always improving, but never complete.